This activity is going to demonstrate how meiosis differs from mitosis. So here we have a whiteboard, and we're going to start off by drawing a circle which represents the nucleus. Each of these represents a chromosome, and so each one has a small white block, which is the centromere, and it also has all of these different colors, which represent the genes. Now in each of our cells, we have one chromosome that came from our father and one that came from our mother. And so we do have the same genes on each chromosome, however, they are different alleles, and so that's why we have the different colors for each. So meiosis and mitosis do begin in very similar ways. So first we have to duplicate all of the chromosomes. So we're going to go ahead and attach a copy onto every single chromosome. So cell division for mitosis tends to be fairly simple. The chromosomes line up during metaphase in the center of the cell. And at this point, the spindles come in and connect to the centromere of every chromosome. And now those spindles are going to separate the original chromosome from its copy, and it's going to move them to opposite sides. So now the membrane is going to form in between, and we're going to have two separate cells. And notice that each of those cells actually has the exact same genetic makeup. Now meiosis is different. Meiosis is the process through which we develop gametes, which are the sperm and egg cells, and for this case we do want genetic variability. We don't want to have the exact same cells, and so we're going to start off the same way with a nucleus, and in here we're going to throw in all of our chromosomes. So the process does begin in a very similar way to mitosis. So first we have to duplicate all of the chromosomes, and so we're going to bring in all of our copies. Now, instead of having the chromosomes line up in a single line like they do in mitosis, now we're actually going to have them line up with their homologous pairs. And so these two will be right next to each other, and then these two will be placed next to each other. So now when they separate, instead of separating between the copy and the original chromosome, they're going to separate this way. Now at this point, there are two factors that play a part in genetic variation, the first being independent assortment. This happens when they line up, and so the way that the chromosomes lined up this way will mean that you will end up with this configuration in the two cells. However, it's possible that they could have lined up in a different way. So, for example, these two could have lined up this way, and so now when the cells separate, you would end up with this configuration for the two cells. So the way that they happen to line up does play a part in genetic variation. Another thing to remember is that when they do line up, they can get a little bit tangled, so they may end up looking a little bit like this, and when this happens, parts of the chromosomes can switch over, and so this is called crossing over, and so they may end up crossing over like this, and so the area containing those genes can be switched over from one chromosome to another. And so we're going to go ahead and remove our genes and switch them over to show how this process of crossing over does change the genetic variability. And the same thing can happen over here. So we're going to go ahead and cross over some of the genes. And so now you can see that we end up with a very different configuration here. So now we're going to bring back our spindles and connect them to the centromeres. And the spindles are going to pull apart the chromosomes. So now we have two separate cells. And notice that they are very different in their genetic makeup already. So they're going to line up once again in the center. And we're going to bring back our spindles, which are going to connect to the centromeres. And now they are going to separate, very similar to what you see in mitosis, where the copy separates from the original chromosome. And so now each of these cells has become two cells, and so we end up with 4 total cells. So each of these cells will be entirely unique. You can see that they each have different combinations of genes on the chromosomes. So if you're a male, each of these will be a sperm cell. For females, it is different. For females, only one will be chosen, and it'll be the biggest one. So it looks like this one is the largest, so that'll be the chosen one, and all of the other cells will disappear. The DNA is within the nucleus, so we're going to draw a bigger circle to show that it is a cell, and inside we're going to add different organelles such as mitochondria, endoplasmic reticulum, and let's say a sperm happens to come to the egg, it'll fertilize it, form a zygote, and that'll become an organism.